Okay, let's dive right into this. Did you know that the perfectly sweet, seedless bananas we eat today are actually the result of careful human intervention? Meanwhile, the famous color-changing peppered moth, that is purely nature's own handiwork. Welcome to this explainer. Whether you're an O-level biology student prepping for exams or just someone fascinated by the living world, understanding this dichotomy really unlocks the why behind the evolution of species. And frankly, it explains why our groceries taste so good.
Here's our interactive syllabus for today. We're going to compare nature versus the human designer, break down the mechanics of both natural and artificial selection, dive into the genetics of ABO blood groups, scale from the micro to macro with sickle cell anemia, and finally, wrap up with some crucial exam pitfalls and a recap.
So, Part 1, Nature versus Human Designer. This is the core comparison that anchors our whole explainer.
And this brilliantly illustrates our starting point. On the left, we've got nature's handiwork, the peppered moth, using its camouflage to survive. On the right, human intervention, today's sweet, seedless banana. We're directly contrasting an organism shaped strictly by survival in the wild with one shaped by our very own grocery store preferences. So let's see exactly how nature's side of the equation works first. Which brings us to Part 2, the mechanics of natural selection, or what we like to call nature's editor. Let's track this chronological flowchart step by step. In Step 1, we start with a mixed population of light and dark moths. Now look closely at Step 2. Notice those heavy, dark arrows pushing down on the population? That right there represents environmental pressure. Think pollution and soot literally darkening the trees they live on. As this heavy pressure forces a survival gradient, we move into Step 3. The light moths get dimmed out, while the dark moths survive, blend in, and reproduce. So nature edits the species based purely on who fits the immediate environment best. Moving right along to Part 3, the mechanics of artificial selection. Enter the human designer. The absolutely crucial difference is right here. Artificial selection is driven entirely by human preference, not environmental pressures. Notice how that environmental pressure from our last step is entirely crossed out? That's because we humans have stepped in. We aren't waiting around for nature to decide what traits aid survival. We're actively deciding what traits are best for us.
So what does that actually look like in practice? Well, we deliberately select highly desirable traits. We take the seeds from only the sweetest apples to plant our next orchard. Or we breed only the absolute fastest horses to create champion racers. The selection gradient here has absolutely nothing to do with evading a predator. It's all about maximizing human utility.
Let's transition into Part 4, genetics, specifically focusing on ABO blood groups.
To really understand blood groups, you need to understand multiple alleles. Basically, this is a genetic system with more than just two allele options. Look at the specific variables popping up here: IA, IB, and IO. These are your exact genetic building blocks. Now, while any one person only inherits two of these, the sheer existence of three distinct options out there in the broader population is what creates our extremely diverse ABO blood group system.
Let's move to and see how this builds in a dynamic Punnett square. Across the top, we have parent AB carrying variables IA and IB. Down the side, we have parent O carrying IO and IO. Watch exactly how these variables merge into the four central outcomes. Every single cross results in either IA, IO, or IB, IO. Because that O allele is recessive, the highlighted offspring can literally only have type A or type B blood. By strictly following these variables, genetic inheritance just becomes a simple, predictable mechanical calculation.
All right, Part 5, Sickle Cell Anemia, where we scale things from the micro to the macro.
Let's zoom way into the microscopic view for a second. Sickle cell anemia is a gene mutation causing red blood cells to deform into a sickle shape. See that structural comparison in the magnified bubble? The normal round cell flows super easily, right? But the sickled cell physically blocks the capillary pathway, which drastically reduces oxygen transport. On a microscopic level, this mutation is a severe mechanical disadvantage.
But let's pull back and look at the big picture. On the left, we've got our microscopic sickled cell, and over on the right, a macro geographic map showing malaria-prone regions. Now, why place these two side by side? Well, because carrying just one sickle cell allele mechanically changes the blood cell just enough to actually provide resistance to the malaria parasite. In regions with heavy malaria pressure, that single mutation becomes a massive survival advantage. So a microgenetic flaw actually becomes a macrogeographic shield. Wild, right?
Almost done! Part 6, exam pitfalls, and recap to help you ace those tests.
Okay, here is the absolute most common exam pitfall: confusing natural selection with artificial selection. Always anchor yourself back to our starting dichotomy. If you see terms like environmental pressure and survival, point your mental arrow straight to natural selection. But if you see human preference or desirable traits, you point that arrow directly to artificial selection.
Now, what's really interesting about this slide is mistake number 2, misunderstanding codominance. Look closely at the variables here. We have a round leaf labeled R and a long leaf labeled L. In codominance, neither allele hides the other. Check out the resulting oval leaf labeled RL. Both the R and the L are fully 100% expressed. It is absolutely not a deletion. It is a complete equal combination.
Let's do a rapid-fire recap to lock this all in. Cement these direct relationships in your mind right now. Natural selection, that equals environmental pressure. Artificial selection, human preference. Codominance means fully expressed alleles, just like our R and L leaves. And finally, the ABO blood group is your absolute classic example of multiple alleles in action.
To wrap things up, I want to leave you with a provocative question. If understanding the why behind evolution completely explains our biological past, how will human intervention through artificial selection and modern genetics shape the evolutionary blueprint of our future? As a quick tip before you go, whenever you're faced with these questions on an exam, close your eyes and visualize the diagrams, the variables, and the directional arrows we walked through today. Because if you can see the mechanics, you can solve the problem. Thanks so much for joining this explainer. Keep asking questions and keep exploring the incredible machinery of life.